just went live.
Oh, okay then. You want me to use a lavalier? Do I need a lavalier?
You can do there if you want.
Hey. Thanks everybody for coming out tonight. Take just a minute to get settled. All right, Paul, you ready?
As ready as we're going to be tonight.
Okay, hey, Tracy Pielmeyer, thanks for coming out to North Central Beekeepers Club. I see a bunch of new faces, which always makes me happy. I understand one of you gentlemen was a beekeeper in Florida.
Yes, ma'am.
For 10 years?
Yes, been over that, for sure.
Awesome, and why in God's name would you move to Indiana?
You can ask my girlfriend. Just moved here in winter time.
She's a crazy woman.
Yeah.
Okay. Okay, well, we are going to put you on our short list to give us some kind of Floridian info at some future dates. So love to talk to you about that afterward.
I'd love to learn about Europe. I'm dying to know what you guys do out here.
Well, we take a little break that you probably never get, so that is a good thing, but it is a little break. So thanks for coming. I'm glad you're here, and you guys are all interested in being beekeepers, is that right? Yay! I think Peg may have told you that we will be doing a Beekeeping 101 in January, February, and March. I think winter is often the time that people are becoming interested, so we will be getting you up to speed after the holidays. So a couple of things. Our guest speaker tonight is Brock Harper, who's the professor of entomology at Purdue University. He runs the Purdue Bee Lab, so we'll be hearing from him in just a little bit via Zoom. Jim is speaking not once, but twice tonight because our very dear Barry McNulty is in the hospital with COVID. So please, all positive thoughts and prayers for Barry and his wife Ann, who's at home. So please be thinking of them. So registration for the Indiana Bee School, which those of you who are unfamiliar, the Indiana Bee School is always the last Saturday in February. It is one of the largest bee schools in the United States. It always sells out at 1,200 people, always. It is supposed to be in Muncie in February, but at this moment, registration has been put on hold pending what will be happening at the end of February. So the schedule is all in place, etc., but we just really don't know, so there is no registration going on yet. Fortunately, Terry will be able to keep us updated how that's going to go. Do I have, let's see, our core meeting will not be a week from tomorrow because we'll all be getting our food coma. And so it would be the next Thursday night if anybody wants to attend core group meeting. Thomas is running raffle again tonight, and then he is going to need to switch off with somebody else to run raffle for 3 months. So anybody interested in helping us out with raffle for 3 months at a time, love to talk to you about that. And anything else? Am I missing anything?
I want to say one thing.
Yes, sir.
Please. Bee school in February.
Yes.
You're going to need to repeat what he says.
I will definitely do that. Come on up, Dave.
It is going to be in Muncie, that's correct. But as soon as registration opens, if you plan on going, register, because it's not going to be 1,200. It's only going to be limited to 400.
Thank you. Very important point. The Indiana Bee School is always 1,200 registrations, except this year. They will only be allowing 400 attenders, and 100 of those will be new beekeepers for the Beekeeping 101 all day. So that means only 300 attenders, non-beginner, if that makes sense. So David is absolutely right. As soon as the registration opens, if you are interested, sign up immediately. So we'll try to get that on our Facebook page as soon as we're aware. Yes, ma'am. Our Facebook page is North Central Beekeepers Club, as long as you can possibly make it. North Central Beekeepers Club.
There's a page and a group.
There is on Facebook, we originally had a page, and that was great, except you all could not interact there, so we made it into a group. So you're looking for the group, North Central Beekeepers Club on Facebook. You do have to request to join, and you have to answer 3 silly questions, and that is to keep out bots. And then we admit you to the group, and then you can all interact on that page. I'm sorry, in that group. So please look for the group, not the page. Anything else, gentlemen, ladies?
Feel free to ask questions on the site if you want to get this a little bit more interactive.
Absolutely, and people who are looking for mentors, you know, we try to find somebody in your local area. For you new guys, weather dependent, we do have 2 beehives here on the property. We do get into them before this meeting every month, and we post that on our Facebook page too. Hey, today we're going to get in there at 5:30 or whatever that is. So that's kind of where you would find that information.
Even in the wintertime?
Not in the wintertime. I know you're not familiar with winter, but we don't do that in winter.
How about the dates we're going to be doing new beekeeper classes in the first quarter?
So January, do I need to look at my calendar? Hold on one second.
January, February, March, and then
Right, so
schedule here.
that third Wednesday of the month, the Beekeeping 101 goes on at the same time as the regular meeting is going on. We'll all meet in this room for announcements, and then the Beekeeping 101 people go to the back of the building, and that goes on at the same time as the regular meeting. And we'll try to put together a resource list for people who might be interested. Honestly, Beekeeping for Dummies is a worthwhile book. It just is. It's not the Bible, but it is helpful, and you can find it used on eBay and at least get somewhere with that. Okay, do we have Brock?
We do.
Awesome. So I'm ready to turn this mic off.
Okay, let me switch over to Brock here. Currently, we've got the presentation going. So.
I'll have to do it.
Brock, we're trying to get you here. Give me just a second.
There he is. I see him.
And
I can see her.
there we go.
There we go.
See if we can change. I'm going to try to make you full screen here for everybody. All right.
Will I get, oh gosh. That's excellent. I'm just going to move my screen because now I'm looking at myself talking. Okay. Hi everyone. Thanks for having me, and thank you for, I can see you waving, that's excellent. Thank you for having me, and thank you for letting me present digitally today. This is, given that I get, all of the bee researchers get quite a lot of invitations, so none of us really want to be the cause of any spread of COVID, so this is a nice, safe alternative. I am here tonight to present to you a little bit about what the lab's been up to the last year and what kind of our driving principles are in general. So, am I able to share my screen? Let's see.
Sorry, I'm adjusting the volume.
Okay, actually, I'm not able to. You've disabled it for me right now.
Sorry.
So, maybe as that gets going, I'll just remind everyone, I'm at home right now. I do have a cat that very much enjoys interrupting Zoom calls. So there will almost certainly be a cat jumping up on this table at some point. I'll move out of the way, but just excuse that if it happens. How's that going? Am I able to? No, not yet. There we go. Thank you. All right. So I should be able to do this now. There. Okay. Can you wave if you can see this? I think I can see it, so that means that you can see it. There we go. Thank you. Look at that. Great. Okay. So yeah, thank you very much for having me tonight. I'm going to talk, as I said, a little bit about what the lab's been up to. First, we are social. So that is, we have some social media accounts. If you're interested in following what the lab is up to, the Instagram page is very up to date. That's the real Harper Lab, as opposed to the fake Harper Labs out there at the bottom. And then my Twitter, that's more my Twitter, but I tend to put out a little bit about what the lab's been up to. So feel free to follow us and see what we've been up to. Pardon me. So my lab is really focused on how we can integrate new genetic technologies into beekeeping, how we can make beekeeping a little bit more informative using those technologies. And before I get into that, I'll just give a little overview of what that kind of means from the 10,000-foot level, so just kind of an overview. So I use population genetics, population genomics, to try and reveal information about populations of bees. So every organism has, in every single cell of the body, a long molecule known as DNA, deoxyribonucleic acid. DNA is a molecule made up of individual units that we call nucleic acids. And those individual units come in 4 different varieties: A, C, T, or G. It's just differences in chemistry between those 4 types. But the order of those 4 molecules along a DNA strand, as they're ordered along a DNA strand, essentially tells cells what to do, tells them what to, how to make certain proteins, and generally how to put together a cell. DNA is also a molecule that is inherited, it's passed on each generation. So when a queen lays an egg, that egg inherits half of its, if it's a worker, it inherits half of its DNA from its queen. If it's a drone, it inherits 100% of its DNA from the queen. When I say that we sequence DNA, what I mean is that we've isolated that long molecule from inside of cells, we've done that chemically, and then we've broken it down into its individual parts, and we just inferred what the actual order of those molecules is. So whether they're A, C, T, or G, what the order is. What you're seeing here in front of you, in orange and blue, are representatives from 2 populations of bees. This is just an example, but it's 2 populations of bees that we have sequenced. So each line in there is a single individual. We've got 1, 2, 3, 4 individuals from the blue population. It looks like 5 individuals from the blue population. And you're looking at their DNA sequences. Now what we do, what I do most of the time, to break down my profession in the easiest way possible, is to say that we look for mutations along those genomes that we can compare. So you can see in bold, I've highlighted spots. I've highlighted mutations across the honeybee genome. You guys can't see my cursor, but it doesn't really matter. You can see a C there at the beginning that's different in that individual relative to everyone else, a bunch of T's, some A's, G's, etc. You can see all of those mutations. Those mutations are really, really important. They're important for a few reasons. First, we can use them to identify populations. So let's say I've sequenced the genomes of, let's say my blue population is an Italian stock and my orange population is a Russian stock. I'm not picking on anyone here, I'm just arbitrarily choosing. And what we find is that T mutation is only found in the Russian population. So that's a mutation that allows us to identify a population. If I sequence another B and I see a T there, I can say with some certainty that it's probably Russian. I can also use this data to estimate genetic diversity. When we're talking about genetic diversity of a population or of a species, we're really talking about the number and distribution of mutations in a set of genomes. So what you're looking at here are 2 populations, and you're looking at mutational diversity. So that the orange population has more mutations and more variation in mutations at single sites. So we say that orange population is more genetically diverse relative to that blue population on top. The final thing that we can do, if we get really lucky, sometimes mutations can associate traits. And what I mean by that is a mutation could cause a certain trait. So in humans, we know that there are strong mutations associated with hair color and eye color. Well, in bees, we can find mutations that are potentially associated with things like this example with varroa resistance. So here what I'm showing, and again, this is just a hokey example, but bees which have that A mutation tend to be more varroa resistant than bees with that C mutation. Okay, so these are the 3 ways we can use mutations. We can use them to identify, to look for mutations that associate with traits. We can use them to estimate levels of genetic diversity, and we can use them to identify distinct population groups. I hope that sounds familiar to you. It probably sounds like this company here, 23andMe. For those who are not familiar, this is a company that will sequence your DNA. They'll take a little cheek swab, actually they take some spit, and they will sequence your genome and tell you a little bit about the mutations you have in your genome, where they may have come from, and traits that they may cause. So essentially, what my lab does is 23andMe for bees. You do 16 and B. So 23, and then humans have 23 chromosomes, honeybees have 16, so we like to say 16 and B. two of these examples today with you. I'm going to look at how mutations can identify populations and how we've been using that information in my research, in my lab's research, and a little bit more about how they can associate with traits. And we'll tie that all back together with what that means for you as beekeepers. So we'll start there. Okay. Before we get into that, though, it's really important to first talk about honeybee biogeography, talk about where honeybees come from. What you're seeing there in front of you, and actually I had to update this a little bit. There is one mistake. If anyone points out the mistake at the end, I'll send you a prize. What you're looking at there in the colored region of the world, that's the ancestral range of honeybees, of Apis mellifera. There are 12 species of honeybee, but of course the one I'm talking about tonight is Apis mellifera, what we'll call the western honeybee. This is the ancestral range. So it's native to the, what we'll call the western honeybee because it's native to the western part of the old world, another poorly chosen name, but regardless, this is its range. And across that range it's been broken up into 5 major population groups, also called lineages. Those are called M, A, C, O, and Y. Each of those groups is genetically quite distinct. And phenotypically, they're quite distinct. They have traits that make them very different from each other. So genetically, if we look across their genomes, we can find a lot of mutations, like that example I showed you at the beginning, where they're entirely different between populations. So if I saw a mutation across the genome, I could say, aha, that came from Africa, or aha, that came from Yemen. So their mutations are very distinct. They're also phenotypically very distinct. If I compare, for example, a Y lineage bee and something like Apis mellifera yemenitica to Apis mellifera mellifera, the M lineage, I will find extreme differences in heat and cold tolerance. Y lineage bees are not very cold tolerant, they're incredibly heat tolerant, and M lineage bees are quite the opposite. They're incredibly cold tolerant, but not so similarly, C lineage bees, these are the Italians, Apis mellifera ligustica, also Apis mellifera carnica. They tend to be a little bit more docile than other groups, and they tend to produce quite a lot of honey. And I'm sure everyone is familiar with the A lineage, the African honeybees. There are actually 12 different subspecies in there, but the one most of us are familiar with is Apis mellifera scutellata, which we'll talk about a little more. But essentially, this is the ancestral population of honeybees. If you see bees outside of this range, they've been moved there by people. And I spent much of my career understanding a little bit of that movement using genetics. And what genetics and a little bit of honeybee records have shown us is that bees tend to move with colonialists. So I've been focusing, most of my work follows Europeans as they move around the world, particularly in these 3 populations. We found the first movement, that when the first colonialists moved, into North, South America, and Australia, they brought with them these M lineage bees. In the case of North America, they brought Apis mellifera mellifera. In the case of South America, they brought Apis mellifera iberiensis, Spanish honeybee. And into Australia, they also brought Apis mellifera mellifera. This tended to be the bee that was used in those new settled populations for quite some time. Really until about the 1800s, the mid-1800s. When beekeepers started to use the Italian system like the goose there, there was a group of certainly one rather famous beekeeper, Lord Langstrip, and others, who made the claim that C lineage honeybees, such as Apis mellifera ligustica, were the best for beekeeping. They were docile, they produced a lot of honey, and by his account, they were quite pretty. So they should be the ones that everyone uses for beekeeping. And this resulted in something called the Great Italianization Event, where beekeepers around the world massively imported bees from Italy. So C lineage bees were spread everywhere, often out, extirpating or removing M lineage bees that were already resident. Fun fact, in the United States, if you were a beekeeper at the time and you wanted to get a pure Italian honeybee, it would cost you about $1,200 of today's dollars. So I don't know many people who spend $1,200 on a queen today. So it's quite an extravagance to have even one Italian queen. So in North America, in the Americas, we also had another major introduction. This is in 1956. when beekeepers in Brazil, pardon me, bee researchers in Brazil, brought over Apis mellifera scutellata. This is a member of the A lineage. Remember I said that there are 12 different species of, subspecies, pardon me, bees in Africa. They brought over one, Apis mellifera scutellata. They crossed it with the local stocks there, mainly made up of C and M lineage bees, for some experimental work. Unfortunately, the cross was rather aggressive, highly defensive. We'll talk about that a little bit more later. And it tended to be very invasive. It actually spread from Brazil, and by 1994 made its way up into North America after passing through Central America. So today, our bees, sorry, before I get to that. These are the major importations that we have really good genetic records of. My grad students actually been going back and doing some fun work exploring what beekeepers have reported importing since, really since the written history of beekeeping in this country. She's done some really amazing work tracking down which subspecies have been introduced. She's found evidence of about 8 or 9, depending on how we look at the records, but 8 or 9 subspecies have been imported to this country since 1622. These are just a few examples here. But just that's kind of just a sidebar to make the point really that our bees are mutts. They're largely mutts of this A, C, and M lineage. So if I were to look at their ancestry, I would find if I were to look at the mutations across their genome, I would find that they originated from the A, C, and M populations largely. Very little evidence to suggest that any of our bees are pure by any sense of that, of how one interprets that. And we'll be discussing that. Our bees are mutts, and we can use this information. We can use this information to really look at the genetics of bees across the country. So we know that there are mutations that allow us to distinguish between A, C, and M lineages, between those source populations. So we can kind of take a 16andMe, a 23andMe approach, and ask how does this ancestry vary across populations of bees, and what kind of bees are you using in your yards as beekeepers? Does anyone out there actually use pure Italians, pure melliferates? What's the difference between a Russian bee and anything else? So we can use genetics to begin asking these questions because we know that our bees are mutts. So in 2015, some collaborators and I put together a means to do this. We identified 91 mutations across the honeybee genome that would allow us to identify the ancestry, that is, identify where these came from. And we created this service. We created a genotyping service that was free for beekeepers, whereby they could, we would send them a sampling kit, very much like 23andMe. They would send us some bees and a survey back. We would sequence it on something that looks like that little chip being held between someone's fingers on those 91 mutations. We'd be able to tell beekeepers where their colonies came from. So where's the source population of their colonies. So I'll remind you, this is the 23andMe kit. I didn't have Google funding at the time. I still don't now, but I'm working on it. So my kit looks a little bit, my kit was a little bit more rustic. This is it here. The kit came with enough sampling vials, which you can see me holding there, complete with bees in this case. It came with enough sampling vials to collect from 6 colonies from any yard. It came with a survey, so that you would have to fill out just a brief little bit of information about where you thought those bees came from. That is to say, what group of bee you think you're using, whether it's, again, a Russian, an Italian, etc. A little bit about your management practice, so telling me how many colonies you kept and whether you're a bee breeder, etc. And then it came with a pre-addressed stamped envelope. And from that stamped envelope, we could pull folks' place of residence. We could pull where the actual samples might be originated from. And it also came with some sampling instructions. So we sent this out. We actually sent 1,600 vials to beekeepers who signed up themselves. This was across Canada. Spoiler alert, I'm Canadian, and this is a Canadian study. I'll talk more about how we're doing this in America in just a little bit. But we had 1,600 people sign up. We sent out all 1,600 of those sampling kits, and we had a return rate of 52%, which at the time I wasn't too pleased with. However, it turns out that beekeepers across the country, again Canada, are actually mandated in certain parts of that country to fill out mandatory surveys. And those government mandatory surveys have between a 2 and 10% response rate. So to get a 52% response rate for beekeepers was stellar. He gave us 800 samples. only did we get samples back, but we also had some wonderful photos and stories. This is a gentleman in Kelowna, British Columbia. He actually took some images of himself sampling for us. So I think he's probably bragging in this photo a little bit about how productive and temperamental his colonies are. But there he is sampling for us. Here's some folks in northern British Columbia. These are actually some student beekeepers at the University of Northern British Columbia. And they were learning how to beekeep and also taking part in this study. Blog posts were written. This is one from Nova Scotia. We had some really lovely and wonderful letters come back. I think my favorite is this one. This is a gentleman who wrote a blog post for us, but he found it difficult to put bees in the vials that we sent, and rightfully so. They're tiny little vials, and we've updated since. If you're wearing gloves, it's a little bit of a clumsy effort to do that. So he actually came up with a method. He came up with a method to collect bees from colonies that involved chopsticks, a funnel, Scotch tape, and a sampling vial. He filmed himself collecting bees for us, and he put it to music. The music is, wonderfully enough, it's Chopsticks by Liberace. And you can actually watch it on YouTube. It's quite a wonderful piece. So seeing a man catch bees with chopsticks. Okay, so here's Canada, most of Canada. You're missing some of the territories there. Each one of those red points is at least one sampling location. We have quite a lot of clustered sampling locations. And at this point, this plot highlights quite nicely that we had pretty much every major city in Canada and certainly every major agricultural area. 800 spots in total across there. So great spread across the country in every province. And one territory that's up by where Alaska is to the left, the top left there. That's the Yukon Territory where there's a red top. So excellent distribution across the country. All of those 800 samples came back to the lab, and they were processed not by me, but by a really excellent team of undergraduate students here. This team of undergraduates would get the sample back. They would take out all of the metadata from those surveys. That is, they would take the survey data that beekeepers sent, put it into a digital format for me for us to process downstream. They would also take the bees, put them into new label tubes, extract DNA, and send that off for sequencing. So a really wonderful, excellent group of students to work with. Right. So each one of those 800 samples was then sequenced at 91 mutations across the genome. And what we were curious about was the ancestry of those samples. So ultimately, what we were asking is, where did those mutations come from? If I ran this kind of analysis on a bee that came from the C lineage, that is an Apis mellifera ligustica bee, I would find that 100% of mutations originated from C lineage bees. So this plot is showing you an example where we have ancestry, the percentage of mutations from a given population. We have colors representing where those mutations come from, and then we actually have the sample across the x-axis. So as I said, if I sequenced a ligustica bee, I would expect it to have 100% of its mutations from C lineage. So it would be a total yellow bar. Same with M, so if I sequence mellifera, and same with A, if I sequence any one of those 12 Apis mellifera subspecies. In a mixed population, we might see something like these examples here, of those 3 Canadian, again, just examples, where we have some mix of C, M, and A. Now, I show this just as an example because, as I said, we have 800 samples. So when you make a plot like this with 800 samples, it looks a little bit daunting. So this is our, what we call an ancestry plot for all of the bees across Canada. Each single little sliver, little bar you see there is a single bee. So we get 800 bees. There are a few takeaways that we get from a plot like this. First, it's pretty obvious that most of the honeybees in Canada are C lineage. That is, they're probably Italian or Carnac. So about 60% of the honeybee genome in Canada is from C lineage. The remainder is from M, it's about 20 to 30%, with a little bit of A. We're going to talk about where that A lineage came from a little bit. But essentially what this shows us is that Canadian bees are really genetically homogeneous. There's not a lot of difference, at least in the ancestry, of those 91 mutations across the genome. What's fun is, what I think is really quite fun with this data set is we can then ask what beekeepers reported buying and reported using in their stocks. Remember, we got them to fill out surveys. Now, by and large, this is where I usually take the poll of the audience, but I'll just kind of spoil it for you. By and large, beekeepers reported buying Italian stock. So largely, they were right. No one uses a pure Italian stock. But we did have beekeepers who reported keeping Apis mellifera mellifera, and seriously so. They had dark bees that they thought were Apis mellifera mellifera. But those did actually not fall up to the Apis mellifera mellifera. They were largely month like this one. We also had beekeepers reporting buying other pure stocks as well, and we have very, very little evidence of that existing in Canada based on this data set. So we kind of come back to the idea of our bees are mutts, they're largely Italian. Okay. Another really interesting thing we can do with this data set is we can begin asking if colonies are northern adapted. And what I mean by that is, do we have differences in ancestry across the country based on latitude or longitude? I mentioned to you that one of the lineages of bees, the M lineage, Apis mellifera mellifera being a representative subspecies of that lineage, is particularly northern adapted. So this is what we call the German dark honey. It's also the first honeybee to be introduced into Canada and the rest of the North. We want to see if beekeepers in northern parts of this country, if their colonies have more M lineage ancestry. So that bar has slightly more, I don't know if you can see my hands, but it doesn't matter, has slightly more kind of black bits to it than the rest of the bar. So is there more M lineage ancestry across the genome for those northern colonies? Ran this analysis for all ancestries, and what we find actually is quite the opposite. We find that as you go further north in Canada, you have less C lineage ancestry, you have less M lineage ancestry, and more C lineage ancestry. Let me say that again because I messed it up. As you go further north in Canada, you have more C lineage ancestry, so more Italian as you go further north, and you have less M lineage, less mellifera and mellifera. I played with this data set quite a lot. I actually thought that maybe I messed up latitude and longitude. It was quite a trial when I was going through this data until we pulled in the survey results. And when we looked at the survey results, it was pretty revealing. Beekeepers in more northern parts of the country, again, northern British Columbia, Yukon Territory, etc., they actually reported higher importation rates. And those importation rates were coming from what we call tropical sources, so places like Hawaii, parts of Australia, and certainly parts of New Zealand, which aren't necessarily tropical, but parts of New Zealand are. So those beekeepers actually had higher importation rates from tropical regions, and we found that imported bees tend to have higher C lineage ancestry. So what we think is going on here is that all those beekeepers in the north are losing their bees in the winter. They're then re-importing those bees from what we call, again, tropical sources. And then losing them again in the winter and re-importing from those tropical sources. So there's kind of a mismatch between management practice and genetics here that we think we've revealed. Kind of a fun and shocking finding. Okay. So finally, I said we talked about the A lineage ancestry across the genome. So why is that there? First, Canada has no evidence of what we call Africanized honeybees, or colloquially it's sometimes called killer honeybees. Canada does not have that. They strictly shut down the border to importation from areas with them, and it's a little bit too cold in Canada for those populations to survive. So what is that lineage ancestry? Well, we had to do a few things to figure out what that was. Our first hypothesis is that it's probably remnant mutations in the genome that originated from initial importations into Canada. In 1882, one of our biggest bee breeders, D.A. Jones, imported thousands of bees from what he calls the Holy Land, the Cyprian honeybee. And Cyprian honeybees are African. They are the same subspecies as some bees in Africa. They're more related to A lineage than anything else, is what I'm trying to say. And he distributed those across the country. So we think we might be seeing some of that remnant ancestry still there. Oops. In addition, Canada has a really rather large group of beekeepers who work with buckfast bees. And the buckfast bees in Canada were actually imported after they were crossed with Apis mellifera saharensis. So that's an A lineage bee, not Scutellata, another subspecies from Africa. So we think we might actually be seeing some of that. If I pulled out the buckfast bees and I compared them to the rest, buckfast tended to have more A lineage ancestry than the rest. So we think that's probably explaining some of it as well. Now this was actually kind of interesting to us, this whole finding of A lineage ancestry, because we realized that we could actually use it to create a test for Africanization. So on the left is another example of an ancestry plot. You have 3 typical Canadian bees, and then an example of what we'll call an Africanized bee. So we essentially reran our analysis on all of the Canadian bees, and then we pulled in samples of Africanized bees from Texas, from Brazil, from Texas and from Brazil. And what we found is that those Africanized bees actually always had over 20% of their genome originating from Africa. So they had a larger red bar than everyone else. So we now use this as a test for Africanization in Canada, this 91 SNP test. And it's a little bit more robust than mitochondrial test, and much more robust than the phenotypic test. So when you actually look at wing donation, what's called the Habits test. We've also repeated this analysis in, this study in Australia, where my collaborators, primarily Nadine Chapman, has found some really interesting patterns of ancestry across the country there. I think most interesting is this difference between feral and commercial colonies. Australia still has a very robust feral honeybee population. They don't have varroa mites. And what she's finding is that her feral bees have much more M lineage ancestry across their genome than typical commercial colonies. So all of that is just from 91 mutations. The honeybee genome actually has 16 million mutations across it. So we really looked at a small smattering of what we could have looked at. So what I've been doing now with my graduate student, Adeline Carpenter, and with my collaborator, Dr. Margarita Lopez-Roque out of Penn State, is expanding this now in the United States, largely the mainland United States. And we're actually looking at whole genome sequencing. So we're actually sequencing hundreds of honeybees from across the country, largely stocks that you work with. We're also taking samples from interested beekeepers, and we're performing some of the analysis that I just mentioned to you previously, along with some additional newer analyses, because we have, of course, more data and more mutations to work with. We're offering a sequencing service to beekeepers. If you do have a stock that you think is interesting and you would like to sequence it, you're more than welcome to send it to me, and we can talk about how to get it sequenced and how to share genomic data with you. We've already sequenced it. I know this is the North Central group, so if anyone's using biters in that room, we've already sequenced the biter. That was the first thing that I sequenced. But if you've got something interesting, just let me know. All right. Sorry. What we're particularly interested in with the data set is trying to identify how many of these subspecies are still present here in the United States. That is, do we still have evidence of Apis mellifera cypria or Apis mellifera syriaca being present in the United States? So we're really trying to dig down a little bit more and see where these have come from. Okay. So that's essentially how we can use mutations to identify populations. Let's, for the last half of the talk here, we're going to talk about traits. I'm really surprised my cat hasn't jumped up on this table yet. Okay. The trait we're going to talk about, we'll talk about a few, but the one I want to focus on is this one here, hygienic behavior. I can see hands. Is anyone familiar with hygienic behavior? Hands up if you think you know. Yeah, okay, a few. Very good. So hygienic behavior is defined as the ability of a nurse honeybee to detect, uncap, and remove dead or infected larvae from the colony. What you're looking at here is an image of hygiene in action. What's happened here is researchers have gone out to a honeybee colony. They found the framed bees around red-eye stage, pink eye stage, and they have frozen a section of it with liquid nitrogen. You take a soup can, you put it over the top of the brood frame, you fill it with liquid nitrogen and fill all of the brood. Put it back inside the colony. Come back 24 hours later and count how many cells are entirely empty. That count becomes the percentage that has been totally removed when you frozen is your hygienic score. It's being scored as a percentage. This column is not particularly hygienic, but it shows you the process in action. You can see totally empty cells. You can see nurses on top of there actually inspecting cells and looking for dead larvae. And you can see some of the cells actually in the process of being opened and actually being removed. So this is hygiene in action. For me, as an evolutionary biologist, and particularly one interested in genetics, it's a really fascinating trait for two reasons. First, it's highly variable within and between species and subspecies. If I compare some of those honeybee stocks I was telling you about, such as C lineage versus M lineage, for example, C lineage bees tend to be more hygienic than M lineage bees. There's variation between subspecies. It's also highly heritable. Heritable means there's some genetic component to it. Sorry, let me back up. It's also variable within species. So if I look within even a single population, there's substantial variation between populations as well. So variation between subspecies, but also variation within a population. It's heritable, which means there's a genetic component to it. It's in fact, on the scale of heritability in honeybees, it's one of the more heritable. It's not something like a Mendelian trait. It's not something like again, human hair color, but there are mutations that contribute to this. There is a strong genetic effect with this trait. That histogram I'm showing you is kind of an example of both of those facts. First, I guess what it is first is hygienic percentage. The number of percentage, of course, being the number of cells that have been totally removed after 24 hours. This is the percentage hygienic for 300 colonies in Ontario, again, Canada. You can first see the extreme variation. So about some colonies remove about 10%, others remove 100% of those cells. So lots of variation. You'll also notice that it's bimodal. There's two peaks. There's a peak at about 90%, and there's a peak at about 60%. And it's bimodal because Ontario has one of the longest running selection programs for hygienic behavior in North America. So those bees in the upper end where the mean is closer to 90%, those ones are from that selected population. So there's heritability, it's selective. Knowing this, my collaborators and I wanted to see if we could identify mutations that cause or contribute or associate with this trait. So we created our own selected populations. This is with Leonard Foster, Marty Marana, Shelley Hoover, and Steve Purnell, Alberta, for most of them, and Vancouver for Leonard. We created our own selected populations. We started with a very large baseline population, hundreds of colonies, all sourced from around Canada, and kept in a common apiary in Alberta. We screened all of those for hygienic behavior. And we randomly, we took the highest, we took 100 of the highest, we took 2 sets of 100 of the highest colonies from each of those and put them into a new population. So we isolated 200 into a new population, separate from each other. All those are selected populations. And then we left the baseline population, that is everyone else that was left there. For 3 generations, we now have 3 populations, and for 3 generations, we artificially inseminated those 3 populations. For the selected group, we artificially inseminated the highest expressors of hygienic behavior with other high expressors of hygienic behavior. So essentially, we created a selected line that expresses hygiene quite highly. For the baseline population, we just did random instrumental insemination. So just grab a queen, grab some drones, mix them together, and then that would be the result. So after 3 generations, we're left with the baseline and 2 selected populations. The figure B is showing you the result of that. So you can see the average hygienic performance of the random samples from the baseline population, so about 70-ish% hygienic. And you can also see from the selected populations that they've actually increased to about 90-ish% between the 2 of them. We can use this data to identify mutations across the genome. I'm not going to go over this figure, because I think that's probably for a different topic, but this one we can go over. So essentially what we wanted to do was sequence the genomes of highly hygienic colonies, like the one on top, and less hygienic colonies, like the one on the bottom, and identify mutations for different between the two. So if you look at, again, you've already seen a set of DNA like this, so we're looking for cases like mutations 1 and 2, where the selected population has an A and a C, and the baseline population has a G and an A. So we can say that the A and C mutations are associated with hygienic behavior. We paired that with some much more sophisticated statistical modeling and some demographic modeling, and we screened the genome for regions that would be associated with this trait. And what we found were actually a few regions. So the honeybee has 16 chromosomes. I'm showing you 8 of them. So each x-axis is just a position along one of those 8 chromosomes, just the first 8. Anything above that red line is a region of that genome that has mutations that are contributing to that trait. So you can see a few peaks, and I highlighted them particularly on chromosome 6. So we found a lot of mutations. Those mutations tend to go, sorry.
Lost.
Not only associated with behavior, but genes that tended to be expressed when bees were developing. So they're genes that are turned on in the brain and help the brain form connections between neurons. So things like this is a gene called ADSIN. It helps form neuronal connections. They also tend to be, the genes we found are likely contributing to this downstream pattern that we see in adults, where highly hygienic adult bees have neurons that are more responsive to uptake. They're just a little bit more responsive in general. So we think what's happening is there are mutations that cause different development in bees, different development in bees, and the development shifts them to be a little bit more responsive to stimuli when they're older. But particularly in neurons at the base of the antennal lobe, making them more responsive to uptake. I'm going to skip that slide and skip on to this. Because we're looking at mutations, we can ask where those mutations came from. And in the case of the mutations we identified associated with hygienic behavior, we found that almost all of them originated from C lineage bees. So you recall at the beginning, I mentioned that C lineage bees tend to be a little bit more hygienic than M lineage bees. And we actually found that across the genome as well, where the mutations that we selected on, the ones that are contributing to hygienic behavior, almost all tend to be associated, almost all of those tend to be from C lineage bees. So when we selected for it, we kind of got the mutations that were not C lineage and selected for C mutations. Kind of a fun population. Sorry, a fun find. And we think this is probably pretty common across most of North America. Because we don't really import a lot of bees. Well, we do import a lot of bees. What I meant to say is because our population is so young, it hasn't developed a lot of mutations of its own. So we think that these ancestral importations probably contribute a lot to the raw material that we're selecting on. So it's not like new mutations are popping up in honeybees here in North America, and those are being selected on. It's probably more likely the case that there are old mutations floating around there that were imported by colonialists and beekeepers over the past 300 years. And those are what we're selecting. So in this case of hygienic behavior, we selected on mutations that originated from Italy. We're repeating this kind of same analysis for aggression within Brazilian honeybees, where we're finding mutations from certain lineages cause aggression. We're also repeating it here in Indiana, where we're looking at the Indiana mite biter to try and figure out mutations associated with that phenotype, and trying to find out where those mutations came from. And I'm going to finish with this one here. This is a really fun trait that my graduate students do, Garrett Slater. We're actually doing the same thing to try and identify mutations associated with male mating behavior. Let's see. Yeah, okay. So males don't really do a lot other than mate. They hang around inside of colonies, and when the time comes, they fly outside of those colonies looking for mating areas. They mate with queens. If they're lucky, they mate with queens. If they're unlucky, then unfortunately they get eaten by birds or they die due to exposure or disease. We really don't have a good handle on how much variation there is in male mating behavior. That is variation in when they leave the colony, how long they're outside the colony, and how many flights they make over their lifespan. So Garrett has been really interested in understanding variation in mating behavior and taking an approach to identify if that variation can be attributed to mutations, and then tying that back to where those mutations came from. He's got a really fun experimental design. Garrett sourced colonies from all over the United States. He actually sourced, there's a typo there. He sourced 3 lines from across the country. He's been housing them here in our what we call common apiary at Purdue. So he's got 3 lines, very genetically distinct, but all available here to you beekeepers in the United States. And he's been tracking them for their lifespan. What he did, when they're very young, when they just emerge from the cell, he takes them and he puts on an RFID tag. It's a little metal cube that goes on their back, and you saw it in this image here. It's that little, just above the green on his back. That's an RFID tag, and he puts those RFID tags on the drones, thousands of drones for each line. And that RFID tag allows him to track the drones every time they enter and leave the colony. So he can essentially do this for the entire lifespan of the drone, monitoring when they come and when they go. During that time, he also collected them. He would collect random samples of drones, and he would assess their reproductive quality. So if anyone in the room has ever held a drone and flipped it upside down, you've probably noted that it will evert on you, that is, it will try and mate with you. Garrett's been doing this with these drones, but he actually collects the semen. He actually records whether or not there's semen at the tip of the endophallus, and if there is, he'll collect it and see how viable that semen is. He's paired that with some what we'll call transcriptomic work, and I'm not talking about that today. We're just going to talk about some of the physical data today. So that little RFID tag that I mentioned is able to collect 50 different phenotypes per male. So from every single male, we know how many flights per day they made, how long those flights were, when its first flight was, when its last flight was. Because the RFID tag has a radio frequency barcode associated with it, we can identify drones every single day. So it's a specific drone that we can track and get all of this information. So first flight, that's like a whole bunch of different data, 50 phenotypes in total that we can track with this. And I'm not going to talk about all of them today. I'm just going to talk about some of the fun variation we're finding. First and foremost, we're finding quite a lot of variation in just when males leave the colony. There's 3 lines here, there's just a box plot. And the point that I want to make is that line 3 down there at the bottom is actually starting to fly. Those drones are starting to fly at age 8, relative to lines 1 and 2, which are starting to fly at age 12, 13. So those line 3 drones are actually going out 4 or 5 days earlier than the others. If those line 3 drones are sexually mature, they're actually getting a 5-day advantage on the other 2. So a pretty substantial amount of variation. We can look at this another way too. Okay, good. So this is a slightly different figure. So this isn't necessarily when they're starting to fly. This is how long those flights are between each of those 3 lines. And you can see, again, compare blue to black here. So black is line 2, blue is line 3. And we find that those line 2 drones are making many, many very short flights. All of their flights are around 5 to 10 minutes. Versus those line 3 drones, which are making really long flights, like upwards of 40 minutes. So those line 3 drones are not only leaving earlier, they're they're flying a lot longer relative to those line 2 drones. And keep in mind, this is all a common apiary. Like environmentally, everything is consistent across these colonies. It's all here at Purdue. It's not like we're comparing bees in Texas to bees in Indiana. This is all in Indiana. So quite a lot of variation just across these lines. And Garrett's continuing looking at these to try and find out the genetics underpinning this. He's actually done some really fun crosses. I'll talk about this. Now, he also looked at sexual phenotypes. So this is a drone that's been inverted, and you can see that kind of brown, coffee-like looking substance at the end of the endophallus. Sorry if anyone's drinking coffee. That is sperm. Garrett collects that. He first notes whether it's present or not, and then he collects it. And he can, from that sample, record its concentration. He can look at how viable it is, that is, how many sperm in that sample are alive or dead. He he can quantify how much they move around on a glass slide or a microscope. And then you can do some more sophisticated things like look at mitochondrial activity and various other kind of fun traits. You can get quite a lot of just individual information from each drone, and compare it, of course, with their flight data. So here we can look, this is just sexual maturity on the y-axis at age 7 and 14, so whether or not they have sperm at the endophallus. And here it's finding actually at day 7, only line 1 is sexually mature. It's really sexually mature, about 30% of their drones, relative to line 3, which is about 10%. You remember those line 3 drones are out there making flights at day 8 when they're probably not sexually mature, they're making flights at day 8 when they're probably not sexually mature, and they're making really long flights too. So really interesting variation at age 7. Similarly at age 14, now again, those line 1 drones are about 60% mature, so 60% of the drones have sperm in the endophallus, versus line 3, where again, it's about 30%. So those line 3 drones are making a lot of flights, but they're actually probably not the most fertile. Similarly, so that was whether there's sperm at the end of the endophallus, whether there's sperm at the end of the endophallus. This is looking at how, what the quantity of that sperm is. Sorry, the quantity, the quantity and the viability. So you can see again substantial variation. This is age 7 and 14, but for 2 different traits: sperm viability, whether it's alive, and sperm concentration, how much there is. So those, again, we can look at those line 3 drones. They're not, this is age 7. Those drones that do have sperm on the endophallus have really super concentrated sperm. Again, a really interesting kind of finding that Garrett's discovered here. And similarly, sperm viability, age 14, line 3 drones, 70% of their sperm is viable relative to about 50% of the other 20. So this is all really new data. This is all actually from this summer and last that Garrett's been putting together. But already finding some substantially I think, informative results that we can follow up on and look at the detailed genetics. So Garrett, as I said, is doing some pretty complicated work downstream where he's taking those low and high populations. He's taking line 1 and line 3, and he's crossing them to create recombinant drones that are of mixed ancestry between 2 of them. He's spreading those out across Indiana, and he's going to see if he can identify, not only, he's going to try to identify how environment contributes to the results that he's seeing, but also to see if he can identify mutations as well. So quite a lot more to come with Garrett's project. I'm excited to talk more about it over the next few years. But ultimately, I hope I've shown you how mutations can identify populations and how we can use those to associate with traits. And a little bit more on some of the phenotypic work we're doing to begin looking at how mutations can associate with male fitness. I think DNA and genomics in general is a very powerful tool for us as beekeepers. I think if we can take a colony, sequence its DNA, we can begin doing some really fascinating things. And things that are not science fiction. We can use it for lineage identification. Stock certification and assisted identification are 2 that I think are really important. If I'm selling you, maybe I won't use that as an example because I don't sell colony. If you are buying a stock and someone tells you it is, I don't want to pick on any breeder, so I'll just say, someone's selling you something and they say it's Italian, genetics can tell you if that's the case, with very high certainty. So you know what you are getting. This is pretty standard in other breeding industries, and it's something that I think is coming towards the industry. You can also use it for selective breeding. By identifying mutations that associate with traits, we can begin pre-screening colonies for whether or not they have mutations that will contribute to those behaviors. So, for example, I can go in the springtime and I can sequence workers in my colonies and see if they have those hydrogenic mutations. If they do, then I save some time for myself in screening them for the actual phenotype. So I'm reducing labor, and labor is spelled the correct Canadian. We can also trait certification. So if I'm selling you hygienic bees, I can also certify that they have mutations that would make them hygienic. Finally, we can identify and preserve local populations. Now, the United States doesn't have much of a remnant feral honeybee population that we know of. Some of my work in the grad students revealed that that might actually not be the case, but we don't necessarily have resident honeybees here. But there are resident honeybees in their natural range, so the C, M, and A lineages, and those populations are actually at risk of extinction and extirpation from beekeepers. For example, bees of the M lineage in Germany were almost entirely extinct because beekeepers wanted to work with Italian stocks. That's the case in Germany. It's also the case in much of in other parts of Europe as well. So we can use genetics and genomic techniques to identify colonies before we bring them into locations and prevent this kind of extirpation through the progression. So I think genetics provides some really powerful tools for us as beekeepers now. It's just one of the many different tools in our toolbox, so knowing the genetics is important. You also need to know a whole host of other things, the location, age, weight, management history, and health of a honeybee colony, but with all of that together, we can really begin looking at predictive beekeeping programs. I have a whole host of people and funding sources to make this work. And, oh, darn, I had a photo there at the end of my lab, but I guess I don't. So I certainly thank my lab for their wonderful work. And thanks to you for your time. I'm here for questions.
How are we going to do questions, Paul?
What's that?
How are we going to do questions?
If they want to say a question, can you hear me okay?
I can hear you, yeah.
Okay, so if they can say it, if he can't hear, then I'll just repeat it.
I have one, actually. Given the uniformity of the ancestry of the bees across Canada, assuming you'll find similar results, or some studies in the U.S. have shown somewhat similar results, do you think that there is any truth to the adage that encourages beekeepers to buy locally adapted stock? In other words, is there such a thing, given the age of the bee population in North America, as truly locally adapted stock?
That's a very good question. There's a few things that one needs to keep in mind when comparing the Canadian study. Up until about the early 90s, Canadian beekeepers did not overwinter their stocks. They would actually kill everything. It was just cheaper for them to do that. So the lack of, the lack of differentiation across the Canadian stocks is not particularly surprising. I should also mention in that data set, the most differentiated stocks tend to be those in the southern parts of it. So Toronto has some of the, Toronto is the southern part of Canada. So Toronto has some of the most genetically distinct bees in the country, relative to newer populations like Alberta and British Columbia. So homogeneity there might be just a signal of the fact of management history. In the United States, early genetic data suggests that there are indeed some genetically distinct populations across the country. We know there's an east and west divide. This is even pre-Africanization. And we know, of course, that there's now a southern divide as well with the encroachment of the Africanized. So we know there's some genetic structure here. As well, the remnant feral populations that we do have, examples like the Arnott forest, the Arno forest, those bees are genetically distinct from others. So we have, in this country, many little pieces of evidence to suggest that there's genetic structure. As well, we, in that Canadian study, didn't really do a deep dive on the local versus national question. The data sets that we've got now in my lab for, again, 16 million mutations versus 91 suggest that there is some genetic differentiation between stocks that are old and local versus, I would say, like new and important. So it's I guess the two-parter answer to your question is comparing the Canadian study is difficult because of those asterisks, those points that I made. And then the second part is really that there's probably genetic structure, and you were asking about breeding local. I'm sorry, what's your second part about breeding local?
The question was, is there truly any value to using locally adapted stock? Is there such a thing, and is it is that genetic diversity in the local environment significant enough to make a difference in survivability, especially over winter?
Yeah, so our early genomic studies, so we had 16 million mutations, suggest that there are enough genetic differences locally that could be contributing to differences. We also have some great studies to suggest that to be the case too. There's my predecessor, Greg Hunt, recently published his last paper on the Indiana mite biter, and that paper showed, I think really nicely, that if you compare, if everything is equal, just equalize your entire operation and drop in queens that are locally sourced, mite biters, and queens that are not locally sourced, put them side by side in those colonies, you'll actually find that the mite biters tend to do a little bit better. Those locally sourced tend to do a little bit better. That's one study that's here in Indiana. I use that as an example. Talk here in Indiana. But you can pick out other ones in Pennsylvania where they found very similar results as well, and Texas as well. So I think there is some value in the breeding and sourcing local. And there's even value beyond the genetics as well. Spreading disease in honeybees is very, very easy. Varroa spreads so easily across the world because we tend to ship honeybees so much. So if Tropicalaps ever gets here and we're shipping bees as much as we are, I suspect it's very critical.
I have one question.
Okay.
Did you say you would be able to genetically recognize feral? Are there, can you find that in your test? If I find bees in a tree and I send them to you, could you tell if they were truly feral?
We'd like to. That's a good question. So can we identify feral bees based on genetic sequence? We are hoping to get to that point, but there's a lot of, so feral in itself is kind of a difficult term, because by definition, any swarm that is outside of a managed apiary is feral. So if I have a swarm from my yard that lands in a tree this year, it's feral. And it won't be genetically distinct from my yard because it originated from my yard. But if we're refining our definition of feral to be a feral population, I should say, we're refining our definition of a feral population to be anything that is outside of management and has been for quite some time, interbreeding with itself, then we do have evidence of that in very specific circumstances. The Arnhem Forest is a case in which there are feral populations that have been breeding with each other for quite some time away from management, and they can do that because it's a huge forest. I very much doubt that we're going to find a true feral, what we'll call feral population in homes of West Lafayette, as an example. We might find them in Hoosier National Park, and that's something that we're looking at. But as of right now, I would be hard pressed to identify if a colony found in someone's backyard is feral, just because of those habits.
Anybody else?
Nothing online.
Thank you, Brock.
Yes, thank you, Brock.
All right. I hope everyone enjoys their evening.
Thank you. Bye-bye.
Bye.
Yeah.
5 minutes, 10 minutes start break here.
And come over and visit the raffle bar.
Yeah, take 5 to 10 here, and then Tim has a short talk, and then we're about done. So thank you, everybody.
I promise I won't refer to the genomic sequence of honey.
So we're doing raffle at $1 for one ticket,
Okay.
$5 to get 6 tickets, and $10 to get 12.
Ooh, you have a queen cake holder.
Can we get
Guaranteed to win.
That's right.
Everybody's a winner.
we got a $5. You the ticket, I keep. What's that?
Did you find this?
Well, yeah, more than enjoy.
I waved back there.
I know, I wave at you. I tried to, it's like, wait a minute, oh my goodness.
So you guys doing okay?
Just one.
I don't know where that name was.
Nice to get out of the house a little bit.
Cindy and I took a
Might be feeding back.
because we usually go up to Jackson Falls this time of year.
Yeah, I can change all the
Both sides. Well, I just got
15,000. So, 25.
You do five.
So the only microphone that was live there was his microphone.
So, we went over there just to check it out.
Because I even the camera,
Right now, there's 11 moving cranes over there. With Thomas.
Yeah.
You have a leg?
But kept hitting the microphone.
Every time he fixed that, it like screwed up.
Yeah.
So does Jim have a presentation then?
He
or-
does, yeah. I don't have it up. I just got this. I put this up for the stream.
So it's kind of cool.
So you know when we're coming back.
6 for 5. Oh, really? Yeah, 6 for $5. Yeah.
Most exciting that we
I was still in for several
Yeah,
weeks.
you have time to clean out your garage, everybody's moving around.
The only reason I even have this one right now is so I can watch that the stream is working. This one is doing the presentation and the Zoom call. And then this is just so I can see what I've actually got coming on the screen.
Can you pull up my presentation so I can see if this is working?
Stories and stuff. The first year I did 2 lives. At 5, yeah. I know. For a whole year it was kind of tough, but when I first started though,
Because I didn't have Barry's talk. And I put notes together up front.
Give a shot.
until I listened, and then in June or July, I took that break. And that's when I.
We haven't talked about mites for a while.
I'm gonna flip back to the, I'm gonna flip back to so the people on the stream know when they're coming back.
And everybody, I told them where I was, gift cards and.
We have anybody watching YouTube?
Yeah, we had 10 people watching concurrently. We've had 28 individual hits.
Nice.
Compared to the number here versus online.
We're Hoosiers for stuff.
But that thing needs to mute
That's almost certainly a problem.
from our typical audio video.
Buyers?
Some nice ones.
Did anybody else have troubles hearing, or was it?
I could tell when I listened to the YouTube stream on my phone in the other room. It sounded like it was coming from a mic in the room, not coming from the laptop straight into video. Because it should be going from here to here, straight out to the stream. All right, you're muted. Hang on. I'll unmute you when I feel like unmuting you.
I've been a school bus driver. I use my school bus driver voice.
But the online can't hear you.
And they can't read my I hear myself now. Okay, let's have a seat. Brock went on a little longer than we anticipated. It's very interesting. I'm always really interested in feral bee populations and would love to know if we can ever find any of those. So Jim, what do you got for us? You ready?
1 or 2. And my mic is on?
All right. Here we go.
Which are you, 1 or 2?
I do not know.
2.
Can you hear me now?
I can.
Oh. Here you go. Got plenty of volume. Okay, well, funny story here.
I prepared this talk in preparation for the meeting, and I showed up promptly at 6 o'clock last week, in the parking lot, and was all set to give this. And I was gently and kindly reminded by my friends in the club that I was an idiot, that I was here a week early. Exactly 1 week early.
Anybody use the I word?
No. I can tell by the tone of your text. So I went home in shame, and I haven't looked at this presentation since then, so we can discover it together. Now, this is for those of you who might be new to the club, a lot of the presentations that I do tend to be a review of material that was published in the American Bee Journal or Bee Culture or in some other literature that's available to beekeepers. And it's kind of interesting to kind of go over what's been said. Some people don't really like reading the journals because it can be a little tough to access, and so we try and talk about what's being published out there to help beekeepers. And this study was actually published almost a year ago in the ABJ, and I thought it was really interesting because as beekeepers we spend a lot of time talking about Varroa, and we talk a lot about Varroa because Varroa is probably the biggest issue we deal with. That's it. I mean, if you can manage Varroa, the rest of it is a piece of cake. And so we do tend to talk a lot about Varroa. And this study actually highlighted a few things that, at least in my own mind, that we had kind of perceived to be true. And this study kind of brought those to the surface and said, yeah, we probably should be paying attention to some of these facts. So how did this study work? It was a really large-scale field trial, and when beekeepers, when they do experiments, when they do research, it all size matters, okay? And the more hives in the more places that are part of the study, generally the more universal the results are. And this one was over 2,000 hives that were located at 11 locations across the U.S. for a period of 22 weeks. 22 weeks is about the beekeeping season in most of the United States. So this was for one entire beekeeping year at a bunch of locations. So they began early in the spring and they ended in the fall. And they were looking at some effects of Varroa mite populations in the hives. And they were doing mite counts every 2 to 6 weeks. It would have been nice if it was completely uniform across the study, but they were relying on some commercial beekeepers to do some of the work. Some was done by grad students, so they had a variety of people helping them across the country, and so they kind of took what they could get as far as the frequency of data. But this whole study was set up and designed to test some new forms of Varroa mite management. Okay? That in and of itself is exciting news because as beekeepers we're always looking for new, better, more sustainable ways to deal with Varroa mites. Okay? And these guys are working with some biological agents which they hope are going to prove to be very effective in dealing with Varroa mites and are not going to have a lot of the drawbacks that many of the synthetic chemicals we're using now often do. But that part of the study isn't done. Okay. This paper is about the control groups that they used. And for any good scientific study, if you want to test the effect of a drug, okay, you have to have a certain number of people that don't get the drug but think they are, okay, and another group of people that are taking maybe the current treatment, okay, and to see how effective the current treatment is compared to the new ones you're testing. Okay. So this study is all about the control groups that they use, as well as the group that was getting the standard typical treatment. And they drew some very interesting conclusions from that. So what's called negative control in a study like this is the group that was completely untreated. They were left to their own devices. Good luck. We'll see if any of you live through spring. Positive control was using the chemical Apivar, which many of us use to some degree or another to manage Varroa mites in our hives. So we're looking at untreated hives versus hives treated with the standard method of Apivar. And then there were these 4 experimental treatments, which I'm not going to talk about right now. It's actually probably been published by now. I'll have to look. So from this study, he came away with a number of lessons. And then that's how the article was published. And so he said, The first lesson they learned from looking at these controlled versus apivar-treated bees was, don't let your mite levels get too high. You never quite recover from high mite levels. This is something I've heard for a while, that it's important to treat your mites and to treat them early. But they came up with some very interesting reasons for that. What they found in looking at the treated, again this is apivar versus untreated, what they found was the number of hives that survived to the end of the study, okay, so it survived 22 weeks. Only 38% of the untreated hives survived. 73% of the treated hives survived, which consider that to be a great result. But it was considerably more than those untreated. In other words, there's a definite noticeable difference in survivability of those bees that are treated with a mite treatment. Okay? This is through one beekeeping season. If you take this, theoretically, and run this experiment in your mind for 2 years instead of 1, if you start out, if you run this hive set through another season, you're going to have about 5% of the bees left. Okay? So when we tell new beekeepers, if you don't treat for mites, your bees are going to die. Okay? It's generally proven to be true. Okay? Now, you don't have to use apivar necessarily, but you've got to control mites. If you're not controlling your mites, your bees are going to die. Your bees might die even if you do, but they're definitely going to die if you don't do something. Okay? So the positive control hives in this case had apivar strips placed in the hive for 8 weeks at the beginning of the study. So they only had 1 treatment for the whole season, okay? Which is less than most of us do, and which is probably why most of us get higher survivability rates. They all had about 2.3 mites per 100 bees, so that's what we call a mite count of 2 or 2.3 at the beginning of the study. And the survivability of those that weren't treated was less than half of those in the treated colonies. So here's the part that I found really interesting here is how the earliness of the treatment affects things. Okay? The lower the mite levels they found were at the time of treatment, the longer the hive was expected to survive. Okay, this is based on some modeling they did from their data. Okay? Just removing the mites, and this is the key point, just removing the mites from the hive, just killing the mites, doesn't remove the damage done by the mites. Okay? And this is where I was getting tripped up in my own head. I thought, if I kill the mites, I kill the mites, it's all good. But you know, it's not the mites that are actually killing the bees in most cases. It is those diseases that are vectored by the mites. Okay? A lot of viral diseases and other illnesses are vectored by mites. In other words, the mites carry them into the hive. And when the hive actually dies, it's dying as a result of, at least in part, these diseases that are vectored by the mites. So if you think about someone who's exposed to radiation, okay, if you remove the radiation source, you haven't removed the damage done to the person. Okay, it's not getting any worse, okay, because there's no more radiation coming in. But the damage that's been done to that individual's body and the increased risk of cancer and all of these other things goes on forever, okay, because that damage can't be undone. Very similar type of effect happening in hives with mites. Okay? They found that treatment was the most effective when the count was less than 2 mites per 100. Okay? That's when you get the most bang for your buck from treatment. Less than 2. More mites, more damage. Higher your mite count is at the time of treatment, the more damage has been done to your hive. I've heard some beekeepers say, well, that mite count was 14, you might as well just walk away. Well, that's ridiculous. I could treat them 3 or 4 times, I get that mite count down. But it doesn't work that way, okay? It doesn't work that way. You have done so much damage that at that point, they've been fatally affected. All right, lesson 2. Don't rely on your neighbor's assessment. Mites vary by location and even by colony. Okay? This is of these 2,000 hives, they were broken up into different areas, and each area, the 2,000 hives were broken up into 11 areas around the country. And in each of those areas around the country, there were numerous yards that were separated by various distances. And they looked at several of these yards that were relatively close to each other. And they used this one, they pulled this one out as an example. It's called UTC yards 1 and 2. Yards 1 and 2 were on opposite sides of a pasture. Okay? There was nothing in between them. They were facing center to the pasture. There was no fence, no hill, no tree line, nothing separating these two sets of colonies other than they were 900 feet apart on opposite sides of the pasture. And yet, the survivability was quite a bit different. Okay? And the survivability was different in large part because of the mite counts in those two groups of hives. Now, there's no real rationale for why the mite counts were so different in those 2 yards, but they were. So just because I go out, and test my bees and I go here to David and I say, hey Dave, it's a great year. I had 0 mites on all of my hives and only 1 mite in 3 of them. David can't say, well, that's great, man, I don't have to treat this year. This is bad. This is a good year for mites. You know, my mites are way down. It doesn't work that way. Okay? So, I said that. Oh, that was the other thing. The mites in those 2 apiaries were bred from the same stock. So the genetic differences that Brock was just talking about wasn't a factor here. They were all from the same stock. The other part of this lesson, local means really local, okay? Even within the same yard, 20 hives in a yard, even within the same yard, the response to treatment can vary a huge amount. Oops. Okay? This is 1 bee yard that was treated with apivar, and the mite counts over time are what are represented, and each one of these lines represents a different hive in that yard. Okay? All the hives were treated the same way, all from the same stock, all managed in the same yard, same treatment put in, same time, the same way. Okay? Follow the manufacturer's instructions. In almost every hive, the mite counts went down. Except for these 3. The blue one here, the purple one, and the black one here. Mites count went up. The mite treatment failed. Okay? The mite treatment failed in that yard. So even if you go into one of your yards that has 20 hives and you say, I'm not going to do a mite count on all of these. I'm going to check 4 of them. This one, that one, that one, and that one. And if they're all low, I'll consider that the mite count worked. Now, you may have to do that because of time and energy and available resources. But the truth is that just like if the doctor gives you a medicine, okay, and you take the medicine, he doesn't say, well, that's done and dusted, we're good. No, he goes back later and checks, did it work? Did it do what it was supposed to? Did it react in your body the way it reacts in that person's body? Okay? There are individual variations that make the response to mite treatments different from hive to hive. And if you really want to know whether you are controlling mites, which is different than treating for mites, okay? Treating for mites means sticking something in a hive or doing some management practice. Controlling mites means actually having your numbers be where you want them to be. If you want to control mites and know that you're controlling mites, you got to count. All right. Lesson 3. Mite treatments don't always work, which we just talked about, and one bad colony can spoil a whole bee yard. This is where the concept, if you've been around for a little bit, the concept of the mite bomb comes in. Okay? They found this sub-lesson, 3A. Mites can recover really quickly. The apivar strips, remember, in the positive control hives, were put in there for the first 8 weeks. Okay, that's a standard treatment for apivar, 8 weeks. Okay? The top chart shows this was the average count out of all 2,000 hives of the mite counts, and shows what happens after 8 weeks when the strips were removed. Okay? At 24 weeks, that mite population had bounced up quite a bit. Okay? That's the average for all 2,000 colonies. The bottom one shows that in various locations, this is two different yards, New York, state 1, Texas, state 2. You know, the Texas population, the mites stayed relatively low after the treatment was taken out at 8 weeks. Texas stayed low. New York population went crazy. Again, if we don't count, we don't know. Okay. So the quick recovery of these mite populations could also be due, though, not to just the mites recovering in the hive from the treatment, but it could be from mites coming in from other hives. Okay? So, Treating isn't the same as controlling, okay? If you have mites from other colonies that are coming into your apiary, where your bees are all nice and squeaky clean and treated, okay, your bees are going to be just as dead as if you didn't treat, okay? Dead is dead. So it's not just enough to test your bees and, or to treat your bees and say, I'm good, okay? You got to stay on it. You got to watch, because you don't really know what your neighbor is doing. And if your neighbor has dirty hives, you're going to be sharing into some of that, okay? This study found that 5% of the hives had more than 5 mites per 100 after treatment, okay? Just across the board, okay? 5% of the hives they determined had failed treatments. So that's, that's 1 in every 20. 1 in every 20 treatments failed. That is a little bit higher percentage than I'm entirely comfortable with, just taking for granted. So here's an example of 1 yard, and this was kind of interesting. This was a separate study, and this was one where they looked at overwinter populations. So in other words, from fall through the winter into the following spring. There was only 160 hives in Alberta. Apparently, Canadian bees are more susceptible to study than American bees, from what we've heard tonight. They looked at it from August to April, and all of the hives were treated with formic acid at the start of the study and had an average of 2.3 mites per 100 bees. Okay, so everybody was treated the same. Everybody at the start of the study had the same number of mites. Okay, in treatment A at the initiation, here, these numbers that you see in these boxes, first of all, these boxes represent individual hives and 4 hives on a pallet, okay? So what you're seeing are pallets of hives here. And these numbers in here represent the mite counts, okay, at the beginning of the study, after they've been treated with formic acid, okay? So these have all been treated according to the manufacturer's directions, okay? Anything, if you see anything jump out at you? We got a problem here, okay? But this beekeeper had a lot of confidence, theoretically, in his mite treatment. So he didn't do any mite counts after he was done treating, okay? These 2 are bad, okay? These over here are not good, okay? They're just not as bad as the really bad ones, okay? By week 4, when they went and did another series of mite counts, this is what they found, okay? These guys here, these are already dead. I mean, they're not literally dead, but they are dead bees flying, okay? And if you look, you start to see the spread, don't you? You see the orange boxes appearing, okay? Next to the red ones, you see ones that were white before are now, curiously, this one got better. That is not necessarily from mites rebounding from a treatment. In other words, this hive that had 2 all of a sudden didn't generate 8 necessarily because it failed. It now has 8, most likely because these hives here are sending out bees with mites on them that are going to adjacent hives and bringing mites in. They're importing mites from these other hives. And then April, after the overwinter, the black boxes represent the dead hives. Okay? And you can see what happened here. This was your trouble spot, and this was your trouble spot. And it spread. This whole quadrant was affected. This whole quadrant was affected. This was probably, in large part, preventable. Because if these issues would have been caught earlier in the season, before you were going into winter, at the end of summer, okay, you would have been able to get ahead of this problem here and end up with a spring that looks like that. Okay? Ultimately, 50% losses in this yard. And not only 50% losses, okay, that's terrible. 50% losses is terrible. But you also now have another, 3, 4, 5, 6, I don't know why this one should be red, 7, 29. You've got another 7 colonies that are damaged to some degree, okay, that have a less than favorable chance of making it through the coming season. Why do we have bees that die in the middle of summer? We have the bees that die in the middle of summer, at least in part, because we have bees coming through winter by the skin of their teeth, and they are so enfeebled and weakened that they're having a hard time making it into the season. So, summary of the lessons. Managing for mites is critically important. I mean, we knew that, but it's always nice to see some hard evidence of that. Treating for mites when counts are low is better. That's kind of like saying jumping out of an airplane with a parachute, pulling the ripcord is a good idea. Okay? You don't have to. It's completely up to you. But if you do it, your survivability chances are much higher. Okay? Mite populations can rebound quickly in some cases, sometimes due to how the treatment affected the hive, sometimes due to bees being imported from other hives. And you can't safely extrapolate results from one yard to another or one hive to another, and you gotta monitor your mites. I feel like a broken record. I get up here a couple times a year and I say, you gotta count mites, you gotta count mites, you gotta count mites. It's not hard to do. You can do it in a couple of minutes. It's very simple, and it literally can mean the difference between living and dying for your colonies, especially over winter. All right, that is all I've got. Does anyone have any questions?
I'd say that graph was a good reason to not put 4 hives on the pallet.
I think that you may be drawing an incorrect conclusion from the start.
I know, you don't do that. I know you do.
All right, thanks everybody.
Thank you, Jim. Okay, do I need to wrap up anything? Thanks, everybody.
Stay tuned for what we're going to do in December.
Yeah, so there is consideration of not having a meeting in December just because of everything. As always, we would love to know topics that you want to hear about. And we try to plan at least 3 months ahead, if not a little farther. So if you have suggested speakers or topics or things that you would like to present about, we would love to hear about those things. Feel free to post them on our Facebook group. We always need help with different positions, whether that's the raffle or just posting. We have somebody who's going to write our newsletter briefing for the Beekeepers of Indiana, but there's always stuff you can do to help out your club. So, like to have that. Thanks everybody for coming. Happy to chit chat with you here as we clean up and put everything away. Anything else?
Can you draw the rabbit?
You do.
All right, so we're going to do, are these two together or is it two individual? There's two different.